Okay. Hi, everyone. Welcome to the CIL webinar entitled Quantitative Bile Acid Profiling Made Reproducible, a Transferable Kit-Based HRAM Workflow. To be showcased here are practical strategies and new solutions for reproducible and transferable bile acid profiling utilizing Move Analytical's new MoveKit BA, a product developed jointly in a collaborative effort with CIL. I'm Dr. Andrew Percy, a senior applications scientist and technical product manager at CIL, and I'll be the moderator for today's event. We're delighted to have you all join us. This webinar features presentations from two prominent scientists in the MS omics fields, Dr. Will Thompson and Dr. Caroline Chidley. A few notes before beginning. The structure of this webinar is an approximate 20-minute presentation from each speaker with a Q&A period to follow each, if time allows. Please type your questions along the way to the chat box, and these will be directed to the speaker at the Q&A session as time permits. With that, I'll now introduce the first speaker, Dr. Will Thompson. Will is a co-founder of Move Analytical, a rising software and consumables company that is devoted to advancing and streamlining metabolomics processes. Will's expertise in the omics field is well established and has been exhibited throughout his past tenures. This included his time at the Duke Proteomics and Metabolomics facility as an associate director and research faculty, as well as in his roles later as a principal scientist in R&D and director of life science business development at 908 Devices. Today, he's going to present the results of a new quantitative bile acid assay featuring the incorporation of a 32-plex bile acid standards mix, HRAM data acquisition, and a novel software workflow. The floor is yours, Will.
Thanks, Andrew. Share my screen. Does that look good?
Not,
Andrew, if you could just unmute and make sure we're looking good and sounding good. I can't see you on
not
the
yet,
screen with the
Will.
presentation mode. You don't see it. All right, let me try again. How about now?
Perfect.
Awesome. Thanks, Andrew. Thanks. Good morning, everyone, or good afternoon, wherever you might be. It's a real pleasure to be here this morning. Andrew, thank you for the introduction, and thanks also to Caroline, Dr. Chidley from UMass, for being here with us this morning. Excited to hear her work. Yeah, the title of my talk is here, and I just want to real quickly say that Move Analytical, as Andrew said, is a new company located in RTP in North Carolina. We're really focused on providing software-defined metabolomics workflow with tightly integrated consumables. Today, we're going to talk about, as Andrew said, a new product collaboration between Move, Thermo Fisher Scientific, and Cambridge Isotopes to quantify bile acids in systems with drastic reductions in manual effort and time to result. So that's the focus. That's the focus of our company. And bile acids, the MoveKit BA is our first LC-MS-based product. I want to go back as an intro and talk and tell a little bit of a story. So I'd like to pass praise to much of the work done, before we started on this project, to sort of the pillars that we're standing on. As many of you know who do metabolomics, creating great data sets is no accident. And you need amazing people to do those kind of work. And the two of those people who are really important in my career are highlighted here. The first is Rima Kadura-Duke. She's was a PI at Duke School of Medicine and the PI of the Alzheimer's Disease Metabolomics Consortium. And as many of you know who do this kind of work, the analytical chemists really need great champions and great collaborators, great people with big clinical visions. And Rima represents one of those many people that I was fortunate to work with. The second person I'd like to acknowledge is Lisa St. John Williams. And Lisa is one of the laboratory members. She was a senior staff scientist and came to us at Duke with 30 years of bioanalytical method validation and method experience in pharma. And she really was responsible for the analytical study design, example of which you see on the top right-hand side, which was used to analyze, many thousands of samples. In fact, the largest cohort is still under review, and there was over 5,000 samples analyzed in a longitudinal Alzheimer's study. And really, many of us who do this understand the kind of work that goes into this effort. And one of my favorite things, Rima's very gregarious, and she used to say things like, "Lisa's amazing, a miracle worker." And those kinds of things are great in the praise. It became very clear, though, that much of the data quality was really specifically because of Lisa. So while you can see this definitely as a success, and it continues to be a success, it's also a challenge. So how do we scale beyond? And the problem isn't Lisa. The problem is that reproducible metabolomics depended on having a Lisa. And so while this is a really impactful moment in my career, it also was a very important moment for us to reflect on how things could be made better. One of the most impactful studies that came out of the lab at the time was this seminal finding from one of the ADMC studies, which was that the ratio of secondary to primary bile acids in serum were associated with cognitive decline. This study involved the measurement over 2,000 specimens using bile acids. And it really opened our eyes to the potential impact or reflection of bile acids on cognitive decline. The paper is the most cited study from the ADMC. It's got nearly 800 citations since 2019 when it was published, but really opens our eyes to the emerging landscape. And then, of course, now the landscape has only expanded since then with lots of new bile acid chemistries discovered. However, for those of you who have analyzed bile acids, there are a lot of practical challenges for analyzing bile acids by tandem mass spectrometry. First, you have high background in LC-HRMS by triple quad assays for pseudo MRM. So you get a lot of data which looks like this figure on the left here for LCA, where it's difficult to sometimes find a peak. And if you do, you spend a lot of time manually integrating that peak. Second, there's tremendous number of isomeric species, 2 or 3 or more isomers that are difficult for automated pipelines to analyze. So you have situations like the classical situation here for CDCA and DCA, where they're closely resolved. And you spend a lot of time manually integrating the small peak that the automated software doesn't do a great job of picking reliably the low abundance isomers. So this is a huge sort of time sink towards getting to these quantitative results. And then finally, with this expanded landscape of bile acids, having a lockdown kit that doesn't look for sort of the myriad of other things, that have been publicized by Peter Dorstein and others, looking for this landscape of functional conjugated bile acids is ever expanding. And so this is a real downside to sort of using traditional methods for bile acids analysis. If we take a step back, these are not the only issues with metabolomics, especially, and with bile acids analysis. We expand the landscape from bile acids out to larger metabolomics workflows. There are a tremendous number of operator-dependent workflows and, frankly, decisions that need to be made. Some examples are shown here, and Dr. Chidley is going to share some of her own personal experience, I think, with some of these areas. But just to highlight a few, so sample preparation is typically something that if the samples need to be derivatized or they have very short stability after being prepared, or something as simple, seemingly as simple as plate organization and queue, developing the queues. Those are manual touch points, which are error prone. and time sinks. On the data collection side, if you have a study of, say, the previous, the study design I showed from Lisa, there's 76 study samples, but 114 injections on that plate. And so lots of analytical overhead. System suitability and data quality are usually a kind of a go, no-go decisions. Those take time. And they have, the QA a lot of times on data quality must wait until the initial data analysis is complete. And only then, it may be days until you kind of realize there may have been an issue with the data collection. And then finally, I mentioned before this very large one with peak reintegration. It's a hidden killer, 4 to 8 hours kind of per plate was our kind of running number, depending on the number of specimens and the number of analytes. Highly operator dependent. And then calibration curve curation is another one where, what fit you use and which points you exclude is kind of it's kind of a hidden sort of unspoken source of human variability potentially in the results. So our goal, this feels like kind of like death by a thousand cuts, if you will. From metabolomics, if you kind of look at it from an outsider perspective, they may think, why would I ever want to get into this business? But really, we're interested as a company in turning this into success by a thousand tiny victories, if you will, right? We want to automate as many of these places as potential as possible. So this MoveKit BA is a big part of that. So this is our first LC-MS kit. And what is it? So specifically, this kit includes, as Andrew said, the 32 quantitative bile acids, 16 of which are sulfated. Every one of them has a stable isotope labeled internal standard. In addition, there's 123 semi-quantitative targets that if you run high res, you'll get we've run standards from Biomics, from Sigma, from other places, and we've added them into our discovery library. And so those are semi-quantitative reporting analytes, which you get as part of the kit as well. It's been validated on serum, plasma, urine, and feces. It's about 10 minutes inject to inject. Quantitative dynamic range from about 10 nanomolar to 5 micromolar. Runs currently on Orbitraps and Stellar MS with expansion to other thermo platforms soon. The kit details, the application, so the Move app that you'll see photos from is included. We deliver all of the LC-MS settings, method settings, extraction plates, reagents, QC standards, et cetera, in the kit. So this is kind of the product as it's delivered and ordered from us. The website with the product information just went live a few days ago, and the information is there. You can either grab the web link or scan the QR code. So as I mentioned, all the pain points. So what we're aiming to do as a company is to create scalable metabolomics, which can reduce the hands-on time by tenfold. The ways we do that are kind of these 6 pillars. We have dedicated efforts and specific design efforts in each of these areas that we've put into our product. And we were really focused largely on where I feel like some others have assumed that the lab will handle these things. We really are trying to make it so that there's robust and scalable methodologies that you can bring into your lab for each of these areas. Today, I'm going to focus a little bit on the calibration strategy, on system suitability, and on quality assessment. At ASMS, we have a number of posters. There's a thermo breakfast seminar that we're participating in that you can feel free to attend those. They have details on kind of the other areas, which I won't cover today. So the first point is on calibration. So what traditional multi-point calibration requires space on the plate, obviously, 7 to 8 to 9 samples, and a lot of human decisions. And so the first real thing that we did was to take a look at ways to drastically simplify this approach. So on the left, you'll have a traditional multi-point calibration for bile acids. This is for UDCA. And what you see is that as going through this type of calibration, the user might, in order to basically get a slope and an intercept, a regression equation, the user chooses to do what type of regression, typically needs to curate or exclude maybe the top point, maybe one of the bottom points in order to get the best fit. So what we look to do is to validate how many of these points we could potentially remove. My initial thought was we would decrease it down to a fewer multi-point calibration, or maybe 2 points. What we ended up finding surprised me, as a matter of fact, is that with appropriate controls, you can use single point external calibration. So on the right is the same curve, in the middle is the same curve, shown where we actually leave out all of the points except for a single point. And then that regression through zero allows us to then back calculate the accuracy of the other points. And what we found is that this is a great for the mass specs, modern mass specs have excellent linear dynamic range. And so as long as that point is in the linear dynamic range of the instrument, so that such that the response is associated with essentially the molar response of the molecule, then that regression, that slope, is very close to what you would get for the multi-point calibration, and as it should be for most the intercept is very near zero anyway, and so a single point can reliably back calculate the concentration. Of course, we need to check our work. So in the application of this within our kit, we do this single point calibration, and then we do a multi-point QC. So we still have low and high QC values that are included in the kit, so that it checks the quantitative accuracy at both points both below and above that calibration point. How this is done in the kit is the formulation and value assignment is done in our laboratory in big batches. We assign those using a variety of approaches, stabilized isotope dilution, etc. And then we have these QC batches. We sync them to the lots. And then in the application of these QCs, on the right-hand side, you can see that basically in the application, the accuracy is independently assessed. So the lines are essentially where the values are supposed to be, and in this QC plot, we provide the values as measured, so you can see if anything went wrong with the calibration during the system. We've demonstrated the robustness of this in multi-batch validation, of course, multi-batch it, 3 batches, 3 matrices on the left. And of course, using standard reference material like the SRM 1950, comparative performance tests, both to published reference values from other laboratories, and then published values from competitor kits. So we're very happy with the performance. We feel like this approach for this, in this experiment, we're getting the same values, nominally the same values as other labs. And we expand the coverage because of the expanded target list. We expand the coverage beyond what is otherwise commercially available. For the rest of the talk, I'll be sharing just a few anecdotes from what we've run an early adopter program we call an EAP, sort of to test user reproducibility and feedback. We distributed kits and samples and software to a variety of labs, and then sought feedback on the performance and on the approach. More on this is going to be shared at the ASMS and at the Thermo user meeting. Just one quantitative result to show here, which is the accuracy assessment across 6 labs, 7 different instrument platforms for the QC high is within the range of what we would expect from a kit with highly experienced users. And I will note that these were all first time. This is the first time in all these laboratories utilized the platform. The last few minutes, I want to share sort of our vision of how we would hope to change the expectation for your metabolomics workflow, specifically around the SST and the real-time data checks. So all of us are familiar, if you've run system suitability testing, to looking at the chromatograms from a test mix, and you look at something maybe along the top here, and you make a judgment call. Through evaluating the hundreds of SST runs across the instruments, we've boiled down the parameters critical for this to the fewest possible metrics, and then we deliver a dashboard in our software which gives you kind of go, no-go criteria for each of these metrics, so that we would know that immediately that the one on the top left is good, the one in the middle is borderline for this sulfated resolution, and the one on the far right is a failure for several different metrics. This removes the inter-operator and inter-coffee, finally, variability. Joke works better when you're live, but decisions for this SST kind of eval. Um, we also want to, sorry, I was hitting the wrong arrow here. When we get to the quality real-time quality assessment, this is where we feel like really shortcut the sort of turnaround to data assessment. So traditional metabolomics is often forensic, meaning several days may go by before you get kind of a readback of what the data quality was. And using strict metrics, we're taking the chromatogram on the top, and for every run on the instrument, the software sits on the instrument and is running, you're getting a plot which gives you intensity readback, migration variability or retention variability readback, and then critical pair resolution on a run by run basis. So we're really trying to push towards this quality control being operational. So if things are going wrong, you find out early. Of course, science isn't perfect, so when you find out is really the key here. So here's an example from a laboratory in this study who was using an older column, and the normal result is shown on the left, and then the results shown in the top are what was found in the data set on the right. And so what we're seeing here is this is a failed data check. There was no additional, operation required for them to know that chromatography for this particular sulfated species, which is kind of like a canary in the coal mine, if you will, for this separation. It's the most difficult critical pair to separate. This is the one you can see that these failed. So this is something that you will know right away, and then you can take corrective action. A great example of that was from Zach Hall at UC Davis at the West Coast Metabolomics Center of how to use this at the next step. So he had a borderline, what we call kind of a borderline data check with a few kind of sporadic potential failures. He chose to kind of move forward, and then in the data analysis step, which in this case took just 5 minutes, click the button, automated data processing. He saw a couple of things. He was initially suspicious because he thought, in his first sample prep, something may have been awry. And he saw that he had one outlier on the study on the one of the urine samples, but even more importantly, he saw that he had his QC values did not lie along the lines where he expected, and so he knew right away he needed to reprep. So he said, "This is a 1 to 2 minute decision. I know I needed to rerun." My last data slide is the performance on automated peak assignment. And so in this case, no expert needs to waste their time in correcting peak assignment errors. So despite the fact that nearly all targeted software struggles with these kind of critical isomers, James Campbell on our team has spent a tremendous amount of time in coding, understanding what you do. So elution order, peak shape, migration, mass accuracy deviations, so that we can understand that, the peaks, they elute in order, which ones should be selected, and the sort of the Move App peak assignment correctness. So across all 6 labs, the 374 kind of runs, for 5 out of the 6 pairs, we got these assignments 100% correct. For this critical pair, as you can see, you start to fail. We did miss on 10%, but those were the ones in the previous example I showed that failed the data check. So when it's going to be possible to integrate these correctly, you'll know right away. And the best quote of this was from Crystal at UNC. Just said, in her experience, the app assigned 100% of the peaks correctly. I had to do 0 manual integration, and this was for a study, a urine and plasma sample she was running after the EAP. So finally, these conventional workflows are slow, they're difficult, and sometimes irreproducible. And what we're aiming to do as a company is address all 3 through improvements in sample prep and processing, improvements in workflow baked into software, and fewer human decisions and more consistency throughout our products. MoveKit BA is the first one that we have worked on, and we're super happy to have been collaborating with Cambridge and Thermo on the development of this product. We hope that you all will want to give it a try. We invite you to reach out to us after the webinar if you'd like to. And importantly, we have Caroline Chidley here. Dr. Chidley is a user, and I think she's going to share with us more of kind of exciting how this can be used and how bile acids are used in her lab. Thanks, Andrew.
Thank you, Will, for that very informative presentation on the new product and its accompanying software. Definitely seems like the capabilities of Power can help overcome many of the challenges we face in the metabolomics field. There is one question that was raised from the field. The data analysis looks like Skyline. Are you using this one?
Yeah, so we use Skyline on the back end, so you do not have to ever open Skyline within our app. But we use a Skyline as a essentially as an audit trail. So what we do is we import the raw data is imported into Skyline. We then we fix the peak assignment errors, import those back into Skyline. And then there's a Skyline artifact, which is the analysis document is left in the analysis folder for you to pick up later. But a novice user does not have to do anything in Skyline, but we leave it there for the experts to kind of go in and check our work.
Sounds great. One more question I'll ask before moving on. How much time does it take to get this kit up and running in the lab, like from box to bench, etc.?
Yeah, and I'd be interested to hear Caroline's experience with this actually. But in most labs, it's within a day. So we deliver the first thing you would run is set the method up and run the SST. Typically, most labs were able to do that the first day, and then they're on to being able to collect their data very shortly thereafter.
Oh, one more question just came in, and then we'll move on, and I'll leave the other for you afterwards. Is 26 bile acids internal standards mix available as a product from CIL?
I'm sorry, is 26? Is that what you said?
26. Well,
Yeah, so this is 32, and it is the standards. That's very important clarification. Yes, we are essentially buying the standards and the internal standards from Cambridge. We repackage them into a much more usable, I think, more usable format. That's good for a plate. And the software and we importantly the calibration standards are not neat. They are a matrix-based calibration standard, so we find that we get better reproducibility across matrices using this calibration approach. But yes, of course, the stable isotopes themselves and the native compounds that we spike in to our matrices are fortunately based from Cambridge, and maybe Cambridge will start selling our kit for us too. I don't know, Andrew, that's up to you.
we can discuss. But yeah, if the IS is solely needed and a condensed set, those are available independently. Well, thanks again, Will, for the excellent presentation. Highly appreciated.